Good morning. I wish I had a glass to ting, but thank you very much all for joining us today. For those that don't know me, my name's Ingrid Scheffer. I'm a bit close now, but I've got a scratchy throat, so it's good that the mics are good. I'm a paediatric neurologist, and I warmly welcome you to today. I want to start by recognising the Aboriginal descendants of the land, and to recognise any of their elders past and present, and any Aboriginal and Torres Strait Islanders in the room. Thank you for giving up your Saturday. I wanted also very much to thank the speakers for giving up their Saturday, and just to let you know that the speakers are doing this out of the kindness of their heart. They're preparing their talks at midnight or 1 a.m. like I was, and they are very kindly coming in their own time to share their expertise and knowledge with you. And I think that that certainly needs to be recognised. I also wanted to thank the Epilepsy Research Centre team. I haven't really warned them, but I'd like them all to stand up now. This is partly so you know who they are. We've got a whole lot of our wonderful research assistants. If you look at the back of the program, you'll see we've got two of our international fellows here, Danique from Holland and Hannah from Belgium, but we also have a number of members of our team. We have Samantha, who'll speak to you later, who's a postdoctoral speech pathologist who works with, many of you will know her. So thank you to our team, and also thank you to the University of Melbourne. In the past, we've had to scrabble together funds to run a day like today, and Krista very wisely put in an application to the University of Melbourne, and they gave us funding for today. So thank you, University of Melbourne. This has also got marking over here, so you should be able to hear me very well. You can see the little instructions here about where the food will be, very important. We've always had a group photo, and we're going to do it at the beginning of morning tea, because before lunch we're all split into separate groups. So we want to make sure everyone can be in it. And that'll be on the steps in the front of the building near all the roadworks, I dare say. And then we have a split session around specific genes just before lunch. Now, for the first time, with this extra support from Melbourne University, we're actually live streaming the event. And so I think it's fantastic you're here in the room. I think you always get more by being in the room, but we appreciate that many of you have a sick child and can't leave that child alone, and so your other half is probably at home looking after the sick child. And hopefully they are into live streaming, and hopefully it's working. We have a special company that called FiveStream, I think, that does this as their day job, so hopefully they're doing a beautiful job. And you can see we have more people online, and apparently I just heard this is the largest online attendance for a conference held by Melbourne University, so that is pretty impressive and speaks to the parent bodies being amazing, I think. The other thing that's exciting about that is they can ask questions. So your questions are really important, I think, because you're often going to ask something other people want to know, but they may have some other questions as well. And our team, Amy and Georgie, are going to be on the bringing up the computer at the end of each session so we can look at their questions as well. All right, so the program, hopefully our beautiful programs put together by Krista are in your hands, and you can see we've got a very full day ahead of us with speakers, mostly Victorian, but Dr. Gill is coming specifically from Sydney for the day. And we have many different types of expertise so that people will share their expertise with you. And I wanted to highlight the last session of the day, which will be chaired by the inimitable Sarah James, who many of you will know already. I don't know if she's here yet. Sarah is at the NBC, I hope I've got that right, journalist for America who's in Australia, and she's also on ABC Breakfast, and she will chair the last session, and you can learn so much from the families. In some ways, I think a lot more than from us, and I've asked these quite exceptional parents to come up and talk to you and to share their expertise with you. All right, so now I am going to try and get this technology working and going to my own laptop. world's a wonderful place, it will work. So there we go. So my job in this hour is really to talk to you about what's this all about? What are these fancy words, the genetic epileptic encephalopathies? And I'm going to proceed through a range of definitions. These words are big and they are confusing. And I'm going to try and put it into English. And then I'm going to talk to you about why we need to look for a cause and why we're calling them genetic, and just drill down on what that really means and the different technologies, because I think at least a third of the people in the room, you don't know the gene for your child's disorder, and I don't know about the numbers of people online with that question. But what can you do to find that, and what other steps should we take? I'm then going to touch very briefly on the comorbidities, which is a fancy medical term for the associated conditions. And you know that all of these children really have comorbidities. And for that, I'm really going to just highlight that they exist and then leave them to the experts. So we have Professor Davey later speaking about sleep. We have Dr. Prakash speaking about behavioural and psychiatric issues and those sorts of things. We have Professor Crompton speaking about mortality, and I'm going to leave that to them. And then I'm going to spend the last bit of a talk about what precision medicine means, because that's the buzzword. There's a huge amount of money being invested in this space. Well, Obama invested a lot. Who knows what Trump will do with that? But this is a very important space for this group of conditions, because they're all relatively rare diseases. Dravet is less rare. The more we know, the less rare it seems. But they're all rare diseases, and we need targeted therapies. And that's where we need to head and where the science is going to take us. So, big words, genetic encephalopathy. I just thought I'd break it down. Apologies if it's all obvious to you. Enceph means brain, pathy means disorder of the brain, and genetic means due to a gene or genes. And in our space that we're talking about today, we know mostly the child has one causative gene rather than multiple genes. There will be some children where it's due to multiple genes, but we haven't even really begun to tease that apart yet. And so it looks like my pictures aren't going to work. Isn't that a wonderful thing? But this is just basically to say, big picture slide, 4% of the population have epilepsy at some time in their life. There are many types of epileptic seizures and epilepsy syndromes. They often have normal brain imaging. So my point is that if you have epilepsy with normal brain imaging, my rule of thumb is it is likely to have a genetic cause. We don't know most of the genes yet, but we know some of them. And if you have abnormal brain with a brain malformation, we know that many brain malformations where the brain hasn't formed right also have a genetic cause. And within this genetic epilepsy space, we have all of epilepsy, really. We have very straightforward focal epilepsy and very straightforward generalised epilepsy, but today, of course, we're going to be focused on the developmental and epileptic encephalopathies. So just to keep walking through what we mean by that, I think one of the most important things around genes causing epilepsy is that it's not all the same deal. And just because you have a gene doesn't mean you know what your child's going to have wrong with you. So we know that one gene usually causes both a mild and a severe outcome. Not all genes, but most genes that we know about have a benign or self-limited outcome and a severe outcome. So if you get a gene test back and it's got a genetic abnormality or mutation, that doesn't tell you the answer about how severe your child's disorder is going to be. And for that we need the neurologists and the paediatricians, because they have to understand the presentation of the child to know which type of epilepsy they have, and we call that a phenotype. So we do the phenotype, the gene scientists do the genotype, and we have to put them together. Now, the gene people would love it to be straightforward. I found a mutation in this gene, that means you have Dravet syndrome, but it's not like that. And that means that it's really important to understand that at one end you have the self-limited or benign variety, and I don't think anybody with that will be in this room. And at the other end, we have this severe group of developmental and epileptic encephalopathies. Oh, trash. So my sodium channel hasn't worked either. Usually things work very well in here. And the sodium channel gene is really just to tell you about ion channels. So ion channels are gateways into the cell, where our bodies are made up of billions of cells. They've got many, many gateways into them, and different ions go through them: sodium, potassium, calcium, chloride, etc. And just to say that many of these severe epilepsies are due to channel disorders, problems with your ion channels. Your gateway is not working properly, but certainly not all. And so I just thought I'd take one example, because SCN1A is the most well-known, and say, well, at this end it causes a very mild benign disorder called genetic epilepsy with febrile seizures plus, where you just have a few febrile seizures at the mildest end and you're fine. It can cause an epileptic encephalopathy called epilepsy with myoclonic atonic seizures. That's one syndrome, but of course it also causes Dravet syndrome. So you can imagine, as a parent, your doctor tells you you've got an SCN1A mutation. What does that mean? And the answer is you need to understand this presentation. And just to give you an idea, if you have genetic epilepsy with febrile seizures plus, or we call that GEFS plus, and you're in a family, you have 10% or less lower risk of having an SCN1A mutation. If you have epilepsy with myoclonic atonic seizures, some people call that DOSA or DUCE syndrome, you have about a 5% chance. But what about Dravet syndrome? Well, there it's more than 80%. So, you actually need to understand the presentation and the gene and all of how this fits together, which is what your neurologist should help you with. Moving on from there, when we see a child, I like to think of them as what I'm seeing in front of me, a complex puzzle about the clinical, the EEG and the MRI and how it all goes together. So it's really important as paediatricians that we know what age it started. Even as adult neurologists, we need to know what age it started if we're going to figure out what type of epilepsy the child or the adult has. What was their first seizure type? What did they come along with? What other seizure types did they develop? And I just wanted to touch for a moment on the two new papers that came out this year of the classification of the epilepsies. So this is the first new classification of the epilepsies in 28 years. So it's been a long time between drinks. And we're using new terminology, and lots of people won't know that terminology, and your doctors may still be using, or are likely to still be using, the old terminology. So when you're hearing this new terminology, we have to teach ourselves to use it. It's easy for the young guys in the audience because they learn straight away the new terminology. But for the old guys in the audience, it's a bit harder, and you'll find that it might be a bit confusing. And just to talk for a moment about the most important of these is that the aim of the new classification was to use words that mean what they say, so transparent words. And when you think about the word complex partial seizures, which lots of your doctors will be using, complex doesn't mean altered awareness, but that's what we mean when we say a complex partial seizure. It comes from part of the brain, which we now call focal, and complex means they're not with it, they lose awareness. And so we've changed that one now to say focal, part of the brain, impaired awareness. So the person loses awareness, and we're trying to use language that is more straightforward. Equally, simple partial seizure was the same as an aura, and that meant you had retained awareness, you were with it through your seizure. And so we have made that a focal aware seizure. So we're actually trying to make language simpler and more transparent so that you can understand and your doctors can use it and everyone around the world can use it. The other part that's important that came out of this new classification, I'm not going to spend a lot of time on classification because I think I want to get down to what's very important for your children. But we have a new framework which I spent about 12 years of my life on, so let's hope it lasts for another 5 or 10. And it begins with the seizure types, and from the moment that any doctor sees somebody with epilepsy, they need to think about the cause, and the medical term for that is the etiology, and then there are these groups of etiologies. You can download these papers from the International League Against Epilepsy website right now if you want, so you can have them to read, but they're very technical. And then we try and move on from seizure types to epilepsy type, and we also try and think about epilepsy syndrome, and we're going to talk a little bit more about syndromes, but they're also quite complicated. And then all along the way, we need to be looking at every child for comorbidities. And to go with this, if you look at the first box, the seizure types, we have focal, generalised and unknown onset, and these are the seizure types. So I'm not going to go through this in detail, but we just talked about complex partial seizure has become focal impaired awareness seizure, and if you're with it, it's a focal aware seizure, just to show you how it works. So back to my little puzzle, and just to put the next piece of the puzzle in there, we look at the development of the child. Was the child ever normal, or was the child normal and lost abilities? And in this group of diseases, most of the children have lost abilities. They may not have been normal to start with, or they may have been, but then they regress and lose abilities and go backwards. What about their examination and their neurological features? What do they show? Do they have skin markings, for example? Do they have weakness down one side? What do their EEG, their brainwave tracing, and their MRI show? And finally, and very importantly, can we find a cause? It's incredibly important to try and find a cause, because once you have a cause, you can move on to the next stage. And I know that, at least half the people in the room have a cause, but the ones that don't, I think it's important every time that your doctor sees your child that you have the question, is there a cause we should be thinking about? Is there anything else we should be doing? And all of this together makes an epilepsy syndrome. So let's try to say this another way. For all epilepsy syndromes, we don't have causes, but space is changing all the time. Every week there are new genes coming out, etc. So it's something we have to keep revisiting. When you start with one epilepsy syndrome, such as West syndrome, you can then evolve to another syndrome, such as Lennox-Gastaut syndrome. So about 50% of the children with West syndrome go on to have Lennox-Gastaut syndrome, and they are both within this group of epileptic encephalopathies. And I found my puzzle has worked really well to try and get across the complexity of this group, and I now have puzzles for most of the different genes, but I'm not going to show you that because that would really put everyone to sleep. It's very hard unless it's your disease. But in terms of syndromes, which I hope I'm showing are different to causes, syndromes, we have a host of syndromes, and they tend to begin in childhood and infancy. And the most well-known, West syndrome is probably the most frequent. Dravet syndrome, obviously, many of you here have children with Dravet syndrome. Lennox-Gastaut syndrome is also a major group, and this one, epilepsy with myoclonic atonic seizures, described by Dose or Deuce, is also quite frequent. But there are other much rarer ones. Otahara syndrome begins in the newborn baby, often in the first few months of life. And this one that begins at around 2 to 10 months is a very nasty syndrome, migrating focal seizures. And of course, the epilepsy aphasia group, which is where children stop speaking. They're also in this group, but they begin a bit later. Now, most of these are generalised or involve both sides of the brain, but there are some that are also focal epilepsies that are in this group, and Rasmussen or Rasmussen would be one of the most well-known of those. Now, something's missing there, so, oh no, my EEG, it's never done this before. This is to show you the EEG of a patient with an epileptic encephalopathy. And what you have to picture, and some of you will have seen this before, is constant epileptic activity. So every second there's a discharge on the person's EEG. And you can imagine if your EEG is having a discharge every single second, you can't learn. And you can't pay attention because your brain has just got this epileptic activity bombarding your brain. And that's really what an epileptic encephalopathy means. It means that the epileptic activity itself contributes to cognitive or learning and behavioural problems in some, but not everyone, beyond that expected from the underlying abnormality alone. So by that I mean you may have a small brain malformation And that in itself might not be so bad, but it can then fire up this process of epileptic encephalopathy, and that pummels your brain and your development stops or sometimes goes backward, or not sometimes, quite often goes backwards, but quite often just stops. And that's what this group of diseases does. So the hallmark is this frequent epileptic activity. You can't have an epileptic encephalopathy unless you have epileptic activity. Otherwise, you just don't have that. And many of these children have frequent seizures, but not all. And many of these children have many types of seizures. And they have this developmental slowing, just slowing, or actual regression. Oh gosh, now I'm going to cry with my beautiful new slides. So I'm wondering whether we can fix this if I ask. You've got that, Georgie, haven't you, somewhere else? Do you want to maybe try and pop it on there and I'll just keep going? Maybe I'll skip these because I can see them perfectly on my computer. And I'm going to come back to that and maybe I'll do that section. Maybe we'll move on to genes. And, actually, what I could do is show you on here, couldn't I? So, can you see that? I'm not sure if the people online... Oh, that's interesting. You see how it just went? All right, we'll see if Georgie can fix it. Because it's a pity not to show you the pictures. I'm going to skip on then to the genes, and they look like they might work. And I played it through actually before you came in, and it worked. And I haven't unplugged it in that time, so that's weird. On my machine. Okay, so we're moving on now to looking for a cause, and why does that matter? And I think sometimes people say, well, it doesn't matter at all. But I have found the cause in some of my 30-year-olds and 40-year-olds with these severe epilepsies. And finding the cause really puts to bed the answers for the family. And I don't think we can estimate just how important that is. I remember vividly long ago that I saw a 17-year-old little guy who walked in. He was absolutely very cute, non-verbal. Yep.
It's very difficult to hear you, Dr. Scheffer, and there's words online that we're having a hard time hearing.
Oh, that's important. It's better if I'm over here?
Yes.
Ooh, sorry about that. Have you got success?
I think so, yeah.
All right, so let's try over. Do you think I'll be that one? It must be. Cross fingers. Yes. Okay, so thanks, Georgie. So now I'm going to have fun trying to do that. Okay, so, thank you. Now, how's that audio-wise? Thank you for making sure you told me. So here you can see that's the very active EEG I was talking about, with discharges every second. And that's really the hallmark of these conditions, that you need to have a lot of epileptic activity together with the developmental regression and slowing. And here you see a little 21-month-old girl with SCN2A encephalopathy. And when I met her, she was having a seizure literally every 2 minutes. And you can see her there in the consulting room having a seizure. And we admitted her directly to hospital at that stage. And you can see that her EEG is full of epileptic activity. It looks a bit different to the one I just showed you, but you can see it is still very active. And this type of constant epileptic activity is associated with developmental slowing or regression. So why does that matter? And you can say, well, what's all the fuss about? Well, the fuss is about the fact that it could be fixable, or there might be something we can do to make a difference. I'm going to speak even more into here. So just to give you an example of that, here you see the MRI scan. This is not this little girl's, but of a child with tuberous sclerosis. And tuberous sclerosis is associated with these, you see these white patches? They're abnormalities of formation of the brain. They're not nasty tumours, they're benign tumours, but they cause bad epilepsy and they cause learning problems. And we now know that if you remove the most offending tumours, the ones causing epilepsy, and this is work's being led by Simon Harvey and Virginia Meissner at the Children's Hospital here, that you can actually stop this whole process and it can settle down and the child can get on with learning. So that is something that's really important to identify early if there's a structural abnormality of the brain that could benefit from epilepsy surgery. Now, that's only a very small minority, but in those patients, it can be a life changer. So that's why it's important to try and control this process. At a more straightforward level, if you have a genetic cause and you might know that gene, then you might know which drugs help and which don't. So Dravet syndrome is a very good example of that. In Dravet syndrome, we know that if you're on Tegretol, you will get worse. It's almost assured. And in fact, it was called severe myoclonic epilepsy of infancy, is the old name, because the children presented with jerking down one side, and people knew that was a focal seizure, and the first drug we use for focal epilepsy is Tegretol or carbamazepine. I try and use the real name and the trade name for you. And we know now the French who described it said, yes, we made them all worse because we gave them Tegretol and it brought out their jerks. And so long ago they've stopped using Tegretol in these children. And so just a simple thing like knowing the right gene can actually tell you which drugs are good. And in this case, we know that topiramate, stiripentol is really good, and which drugs could be making your child worse. So some people say, well, will knowing the gene change it for your child? And the answer is definitely it will make a difference. On the other side, SCN1A this is, there's also 2A and 8A, and Catherine will talk about that later. But we know that some of these same drugs, Tegretol and Dilantin, are really good for those children. And we only know that from anecdotal experience, just children who've responded to high doses. But I just can't emphasise enough that if you get the right drugs, you might help your child's developmental course rather than hinder it. Okay, so I'm going to move on here, and I think that's where we were up to actually. So the point here is this terminology is new. You can use developmental and epileptic encephalopathy. And the difference between these two terms is one is this process I've just been describing in detail, but you may also have a developmental component of your gene. So your genes, say STXBP1, SYNGAP1, these genes can be causing slow development in their own right. And is there actually, can one of my team, whoever's got the lovely little baby, there's actually, I think, a room there that's for babies to go and sit in if you want to maybe try that and just see if that might be good. Thank you. So the point of this new idea of developmental and epileptic encephalopathy is to acknowledge that the gene itself is causing slow development, and superimposed on that you have the epileptic process. And how do we know that? Well, we have examples where the children are not normal to start with, and we know that their gene that's causing their disease is causing slow development. And then you add on top of that epileptic encephalopathy process. And so what matters is that may have something that you can fix a little bit. And if you can fix that a little bit, you can help the child to maximise their developmental potential. And examples of this is the little boy that you see here who has a potassium channel disorder called KCNQ2 encephalopathy. Sarah, who's going to be chairing the panel later, is on, you can see her on Australian Story about her daughter with KCNQ2. And if you watch that Australian Story, you'll see that her daughter is much milder than this little boy here who's 4 years old. So the idea of a developmental and epileptic encephalopathy is that you have two processes going on here, and they're both important, and both can, developmental encephalopathy you're born with usually, you're never normal in your development, and the epileptic encephalopathy occurs at any time. And we're moving now towards the idea of a gene encephalopathy. So I just said that with KCNQ2, so we take it away from this sort of big group, but down to a very specific disease. And then I always encourage you to become a world expert in your child's disease. And thankfully, there are family groups that now help that to happen. So, and so you're right, they're both a while ago. and they both sound fine. So my advice to you would be to photocopy them, send them to Catherine,
All right.
and saying,
What does she want?
It's 06, you probably do.
You know what?
5 years later.
From memory, I'm supposed to do.
Wonderful, thank you, Amy, to the rescue. So the little boy on the left has STXBP1 encephalopathy, and he has a movement disorder. And I'm just showing you a picture of 3 children with different movement disorders. He's got a figure of 8 head stereotypy, we call that. This little girl you can see is much higher functioning, and she has a cerebellar movement disorder, so she has a tremor and she has a bit of a head shake, and she can't write and draw very well, but she's obviously much higher functioning. And then the little boy on the right has got Dynamin 1 encephalopathy, and he has this very severe movement disorder, which actually makes it hard to care for him, because as he's a bit bigger now, he goes like this all the time, and it's really hard for his family to manage him. So we now recognise there are more and more movement disorders associated with these conditions. All right, so I'm going to move on now to looking for a cause, and you've seen this slide for a little while before, but basically I was emphasising how important it is to get an answer for the family. Sometimes mum's been worried she had a glass of wine when she was 8 months pregnant. Could it have caused this? And the answer is no. And I think it really does help you to know that that car accident you had or something was not the cause of your child's disorder. It helps you to look for the associated comorbidities, associated disorders, to understand the prognosis and to guide the management, the right drugs, as I've mentioned. It helps you to avoid unnecessary investigations. So if you're going looking for mitochondrial diseases, we do liver and muscle biopsies. Nowadays, with the gene testing, you could avoid that. It helps very importantly for recurrence risk, and recurrence risk is something parents live in fear of. Will I have another child with this disorder? I have a few families who have 2 children with the disorder in that family, and it is really difficult. One of the mums who's coming later today, I don't know if she's here yet, they've got 2 girls with a SYNGAP1, and when they came to see me last week, they needed 2 carers and 2 parents for the 2 girls. And that life is tough. It's really tough. So the idea about recurrence risk of another pregnancy is critical, and there's some very exciting work that's beginning to change that space. International community, the national communities are important, but the international communities are just as important. And I think they're moving every disease forward at a rate that was previously you couldn't believe it would happen. It's very exciting. And maybe with prevention one day, and that's sort of the goal. So we're also looking at diagnosis leading to precision medicine. So I just wanted to give you a brief primer in the causes of the developmental and epileptic encephalopathies. There are many causes. You can have a stroke as a newborn baby and end up in this space. You can have a brain infection like encephalitis. You can have a very bad birth. Now, when I trained, people thought everything was due to a very bad birth, and we now know that's far from the truth, that often children that are abnormal don't cope as well with the stress of birth. And we know that many of these children have malformed brains, that there was never that, or they may have a genetic condition like this. So the lack of oxygen at birth is much less than we used to think. So genetic causes are much more common, and as I've already mentioned, they can be in the setting of a normal brain or in a malformed brain. Can the rest of the world hear me okay? Do we know that now? Is that all right? Good. Okay, so this is a very brief primer in genetics. I apologise if you know all this. I'm on the wrong computer now. And basically that we are made up of about 1 trillion human cells, and we also have a lot of bacterial cells on us, but we don't need to know about that. And our genome has 3 billion base pairs of a DNA, and they're housed on these 23 chromosomes, and we now know we have 19,000 genes. Now, you might think we're the cleverest animal on the planet, but look at Mr. axolotl. His genome is 10 times bigger than ours, and he can grow a new limb when it falls off, which we certainly can't do. He can even grow a bit of his brain back, which would be pretty handy. So let's go then to our genes, and we all see this on TV, this double helix, but this is our 3 billion base pairs. In every cell in our body, we have 3 billion of these base pairs. 3 of them make up an amino acid, and that is the building block of a protein. And I'm not going to spend too much time on the biology, but here you see a cell. Your DNA lives in the nucleus, in the centre of the cell. It's transcribed to RNA, and then it's translated here into making it into a protein. And these days they can do beautiful fancy pictures of proteins. They look like this. And here's a really cute little protein called kinesin that's a motor protein and is involved in taking things around the cell. And that's the sort of thing we're looking at. So what happens when our genetic code goes wrong and we don't get the right genetic code? Well, there are a whole range of different abnormalities. You can have a major chromosomal abnormality, such as Down syndrome, where you have a whole extra chromosome number 21. You can have copy number variants, and I'm going to spend some time on that because I know there are parents here with children with copy number variants. We're going to spend a little more time on the single gene abnormalities, and we're not really going to touch on the polygenic abnormalities. So I like to make the illusion that if you're looking for a copy number variant, it's like finding an abnormal suburb in Melbourne. So it's like you've blown a whole suburb of Parkville out of the water. It's there, it's gone, it's deleted, or you've added an extra copy of Parkville into Melbourne. And the way we study that is called a molecular karyotype. Unfortunately, it's got a whole lot of names: microarray, array CGH. And this looks at the whole of your genome to see if you've got extra copies or copies missing. And what we might, a SNP microarray, here you see an example of a deletion where there's a bit missing compared with a normal. And here you see an example of a duplication where you have an extra bit missing. But unfortunately, it's not as simple as all that. And that's because we all have copy number variants, every single one of us in this room. And this is the 23 pairs of chromosomes that we have. And on the right, these are copy number variants in normal people. So we all have copy number variants. You can see that these are the positions in which there are known copy number variants that are frequent in the population. And on the red side is a little bar that tells you how frequent this one occurs in the population. So finding a copy number variant is normal. And you need to know, is this one important or not? And so the issue here is it definitely the cause? Is it a well-established copy number variant, such as MBD5 deletion, and we have a family here with that, and I think some more online? Or is it something like Angelman syndrome? And here you see a beautiful little child with Angelman syndrome, where we know that this copy number variant, this deletion, causes this very syndrome. Or is it just part of the cause? Is it one of the players in the cause, but not the whole cause? And that sometimes we find when we know mum or dad have it. Mum or dad are normal, but we know that you have an increased risk of having a copy number variant in a disease group like autism or schizophrenia or intellectual disability. And sometimes we just don't know whether it's important or not. And that's if it's the first time it's been described, or there may just not be enough information out there, and then you're left saying, well, what does this mean? And your doctor says, I don't know. And that's unfortunately the state of the current situation. I'm going to skip these two because I want to move down to finding a mutation in a gene, and that's like finding an abnormal brick in Melbourne. So that's the scale of the problem for each of your children where you know a specific gene. And in these monogenic disorders, we can approach it a number of ways. We can look just at the gene, and I'll again use SCN1A as an example, but that costs about $1,600 to get SCN1A sequenced in Melbourne or Australia. That's a lot of money for one gene. And then there's a process which you can do whole exome sequencing, where you're taking 1% of a genome and sequencing it, or this whole genome sequencing, the whole apple, where you're taking the whole lot. Now, fortunately, we've really moved from the bite to the segment of the apple, because of the cost. And the cost has plummeted in recent years with new technologies called next generation sequencing, and we know it's much cheaper. And then there are many ways you can do this, different ways with the whole genome and the whole exome. So with the exome, you see here, we're just looking at the 1% of the genome being all your genes, that code for the protein that make up our bodies, make up your brains, make up your cells. And we're just looking at this bit. And the reason for doing that is it's easier, it's 1%, and we know that's where a lot of the action is. We know most of the mutations are in that 1%. In the whole genome, you're looking at the 100%, and at the moment that's largely research. And we're doing a lot of research in that space, but the analysis is incredibly difficult, and we need more of these very clever statistical people called bioinformaticians, and they're still trying, it might take them 6 months to figure one out, just to tell you how hard it is. So which test should your child have? Well, every child should have this chromosomal microarray. I don't think there's much space to do a specific gene anymore, and that we are looking towards whole exome sequencing, but that's still costing in the clinical space about $2,200 if you were to get it at children's, with an analysis of the genes that we look at, which might be a targeted exome where you're just doing the genes of interest. Then you might expand it to look at the whole exome. And at the moment, the whole genome is really mostly in research. How do you tell if it's the cause? Well, the problem is that if we do an exome in any of us, we get 22,000 variants. So how do you tell which one is the one causing this severe disease? An easier way to do it is to do a trio. So you do mum, dad and child, and you say, what has the child got that mum and dad don't have? And if you're lucky, you should get between 1 and 5 variants, and you should be able to choose the variant. That works about 20 or 30% of the time. And then we have a whole lot of processes, which I won't go into, about trying to figure out whether a variant is causing the problem. So then you come up with this funny old name, a gene name. The gene names are horrendous to try and learn, as you know, with your own children. And just to give you one example, this is a potassium channel gene. It starts with the chemical symbol for potassium, the word channel. It's a family of that channel called Q, and it's the second member of that. So what does that mean? Nothing to you, let me say, and it takes us a while to understand it as well, but it's the name of the gene that's coding for this potassium channel, and this is what the sort of potassium channel might look like, and this is an ion channel, the potassium ions go through here. But that's still not the end of a story. I always say finding the gene is the beginning of a story, because then you have to figure out, does it muck up the system? Does it stop it working? And that requires very clever scientists who are in this building, a lot of our physiologists, and they actually look at these in the cells, in the animal models like mice or zebrafish and things, and they say, does this abnormality of the gene stop the protein working? And that's really important. And then this sort of work is then the basis of disease groups. And I'm just showing Casey and Q2 because I'm going to Boston at the end of the week for 2 days to their meeting. It's the first time I'm going to actually make it in person, not just do something online. Very excited about that. But that's where the families get together with the scientists and the clinicians and talk about how can we move our disease forward, how can we find a targeted treatment for our disease. And I really encourage every family group to do this. It's hard when you're dealing with such a sick child to have the wherewithal to make this happen, but inevitably with the world stage, this is happening more and more. And just to say, there are many, many ion channels and also many types of channels, and they're all being implicated in causing different diseases. Most of the patients that we see have a new gene change in them, which we call a de novo dominant. So new in that child, dominant means if you have it, you have the disease. But you can have other forms of inheritance, such as 2 bad copies of a gene, or one that follows X-linked, which is one of the X chromosome, which XX is a woman and XY is a man. And then of these diseases, about 4% of the children have a copy number variant that is the whole cause, like the Angelman one I was talking about, or MBD5. The problem is a little more complex because one gene causes many epilepsy syndromes, as I told you about with SCN1A at the beginning, but also one syndrome causes many genes. And this is just a figure from a paper that we wrote in a review a couple of years ago. And these are neurons, and I'm just showing you those so you can see this is what our schematic is meant to look like. And if we drill down on the schematic, you see each of these little symbols represents a different gene. And that was 2015. So 2017, there's probably another 50 or so genes to add to that list. And they have a whole range of mechanisms they work through. So it's not just the ion channels that we talked about, but a whole lot of different proteins that are involved in these diseases. So what about the children where it's not solved? What's going on there? Well, firstly, they may not have had 2017 testing. Secondly, they may have an abnormality that's not present in the exome and that we need the genome. It could have been missed by the technology. It could be not genetic, so we know that there are acquired causes, but we don't have a lot of knowledge about them. So this is the testing that we should be offering nowadays. Now, I'm just going to talk for a moment about the comorbidities because I mentioned that these disorders don't come together. They usually come with intellectual disability or at the least learning problems. They often have autism spectrum disorder. I would say 40% or so of the patients with these disorders have this diagnosis. And genes, we now know that code for one of these disorders may code for the others without it. So SCN1A can cause autism without epilepsy. It can also be that they have malformations of the brain, and we know that some genes are more likely to have subtle malformations, and then we have to go back and look harder for them. And here are a whole list of comorbidities. Most of these will be being addressed today. And this is just to show a picture of Molly, who died a few years ago. Her mum might be here today. I'm not sure if she's in the room yet. Oh, yes, she's waving at me, Kelly. And she runs the Molly Foundation. And Molly died about 5 years ago. And I'll be talking about Molly's cause of death in the SCN1A section. But just to say that death is something that we're all terrified about, and understandably so. And I've asked Professor Crompton to address this very directly with you. So just in the last few minutes, I wanted to talk about what precision medicine means. And there's an urgent clinical need for this. And if you like, it starts with the genetic abnormalities, and then our scientists have to sort out the disease mechanisms and model the disease, often in the cell. And we're increasingly asking you for skin biopsies because sometimes they can't look at how those proteins work in the blood, or often, and they want to look in the cells to see how those proteins work. And then we might be changing the skin biopsies, growing them into what we call stem cells, so going backwards with them and then going forwards with them to make neurons or nerve cells so we can look at what's going wrong. Because unless you get down to that science, you're never going to be able to sort out a targeted therapy. And then they also develop the animal models like the mice that I was talking about. And then we need to work together with the scientists to develop new therapies. We can repurpose old drugs, and I'm about to tell you about that example, or find new ones, which is a very exciting space. And then the doctors, your doctors, and we need to work together to bring it to you. And that should be in proper trials, not just, hey, this is whiz-bang, this is going to work, because we don't know if it will work. Then once we show it does work, we need to bring it in and implement it on the world stage. And again, families are fantastic for that because you can use social media, etc., to get awareness out there. So this is what we call precision medicine, and at a simple level, a gene diagnosis might put you onto the right drug versus the wrong drug, might tell you to look for a malformation. It may be that we can get a drug that targets a pathway, not just your gene, but a whole group, and that might be more cost-effective for the society. And then the functional effect of a gene mutation, reversing abnormal function. So when people talk about gene therapy, they talk about a defective gene, and then you introduce a new gene to fix the problem and the cell function goes back to normal. But in some of these disorders, you have an overactive gene. It's got a gain of function, not a loss of function, and then you might want to stop it and repress it. So the processes are very complicated and have to be targeted. I was going to give you this example, but I think I'm not going to in the interest of time. But I'm going to tell you the short answer to this example, which is this was a targeted therapy, a repurposed old drug for a very severe disorder called KCNT1, causing severe autosomal dominant nocturnal frontal lobe epilepsy. But some people here may have a child with KCNT1 with an even worse syndrome called epilepsy of infancy with migrating focal seizures. And that potassium channel is modified by an old drug called quinidine, which works on the heart. There are lots of case reports now of quinidine helping children with these very severe diseases. So my colleagues, Saul Mullen and Patrick Carney, have just completed a study of quinidine in 6 of my patients with the less severe form, which is still very severe, of autosomal dominant nocturnal frontal lobe epilepsy. And when we did it in a blinded manner with a placebo in hospital counting seizures, it didn't work. So the point is there are lots of anecdotes and people say, hey, this is the best thing since sliced bread, but the placebo effect, which Dr. Gill will touch on, is huge. And I just want you to point out that we really need proper trials. So just to conclude, we need new therapies for these severe disorders. We need to understand the presentation, the phenotype of the child. We need to understand, is the gene change really an abnormality or we don't know what it means? And that will already start sending us in the right direction for choosing the right drug. With old drugs, there are some new tricks that we can find with this quinidine, for example, but then there's also the very exciting space of gene therapy, and there's some wonderful things happening in that space that aren't quite in the epilepsy stage, but are just beginning to affect muscle disorders, etc. Genetic counselling is incredibly important for families, for you as parents, but also for your children when they grow up, your other children. They should have formal genetic counselling. I've already mentioned a few times the family groups are incredibly important in supporting you and in taking your disease further forward. There are many different epilepsy syndromes and many different genes, and we need to understand the spectrum of disease. And in America now they're calling these natural history studies, but it's really what we are all doing together every day as we're going through your child's life and trying to understand what's going on for your child. And the way forward is this precision medicine. So I just want to finish by thanking our amazing team at the Epilepsy Research Centre at the University of Melbourne. We collaborate with scientists from Adelaide and from here from the Florey. We're all a tightly integrated bunch. And our team that you've seen today are all giving up their time today to help us. I wanted to mention for a moment Heather Mefford and her team in Seattle, because they've done a lot of the discovery of the genes for you with us. And particularly I wanted to thank you all very much for being here today. Thanks very much. Now, this talk was just slightly longer than it should have been, so we only have 3 minutes left. So would somebody like to come and run around for us a bit? Thanks, Amy. Are there any questions? I will be around all day, though, at the back there. We might need you to get we'll have to have some running happening, I think, girls, because we've got 3 minutes.
We have questions here.
Oh, and we have some online. Yes, I'll just take the one in the room.
My son has a SCN1A gene, has quite a typical Dravet phenotype. We were diagnosed by a specific gene sequencing. Is there any value in going back? We've given our DNA to you, but is there any value for him in going back and doing the whole exome sequence?
No. I mean, if your child has the right presentation and has a gene abnormality that is the right thing, I think there is a value in checking if you and your husband have it, because I think it's very important if one of you has it, then it's not the whole answer because you don't have Dravet syndrome. But if it's new in your child in the setting of Dravet syndrome, you do not need to do any further testing. Okay, so one question here from Rana is that does a truncation mutation necessarily mean a worse outcome? No, it doesn't. In Dravet syndrome, half the children have truncation and half have missense. If you look at the literature, there are small differences, but I think they're immaterial. So, for example, your absence seizures might begin 2 months earlier. I don't think that means much in terms of the big thing. Now, there was another question here. Where are we?
Our son has an inverted duplication of chromosome 15.
Thanks.
He started having epilepsy when he was 22 years old. He's 26 now. Clonic was his first seizure, and now he's having absence and atonic seizures. Do you have much experience with that abnormality?
It's supposed to have a high frequency, that particular abnormality has a high frequency of epilepsy associated with it. I have only had one patient I've looked after extensively who's about 25 or 8 or something, and he's had horrendous epilepsy for a long time. So the fact that your son's began later is unusual, I would think, but also it sounds like what you're saying, that he does fit very clearly into the epileptic encephalopathy group, where he has the multiple seizure types and the severe outcome. And I think, is there another question here? Yes.
Hi. Our daughter's got NPC1. We don't know anything about it. Is that generally associated with epilepsy, or is it?
I have to say I don't know anything about that gene, so I can't answer that. Maybe Catherine will, but where's Catherine? Are you here?
Niemann-Pick, yeah.
No, you can talk to it.
Sorry. So Niemann-Pick type C is a genetic condition, sometimes considered a metabolic condition because of the type of chemical processes affected. There are quite a lot of patients reported, and most of the reports don't specifically focus on the epilepsy. They often focus on some of the other features of the condition, like development, and like things like drops that are falls to the floor that can sometimes happen with emotional stimuli and things like that. So it's one of those things that's not super well described, but there are people working on treatments and things like that for it.
Thanks.
And I'm not going to take any more questions because I'm over time, I think 1 minute, but there are 2 things from around the world. Thank you, listeners. And I think they're both really good questions. So Karen from the United States has said, is a syndrome a clinical diagnosis? Yes, it's an electroclinical diagnosis. And a gene encephalopathy is also a clinical diagnosis. And I hope I've shown you that they're related but not exactly the same. So many syndromes tend to have more than one gene, and genes may have more than one syndrome. So they're very important but different concepts. And the second question that I wanted to, Teresa from Australia, really good question, thank you Teresa too. Should adults with uncontrolled epilepsy with intellectual and psychiatric disabilities be checked for a genetic problem? Absolutely yes is the answer there. And often the adult physicians aren't as tuned into genetics. As paediatric people are great at it, it's our day job, but adult people are not as good. And so if you have an adult child that has a disorder, you should be into your doctor and saying, what can we do to figure out the cause? And I'm going to stop there. And thank you, Amy.